Hello, this lecture is for RCBC general biology students, but we certainly hope others will join in. This is a continuation of our discussion on biotechnology. During our Collaborate session, we discussed recombinant DNA technology and how we could utilize that technique and create what we call a recombinant protein, such as insulin. It is a human insulin protein that is created in E. coli. So that gives us an example of how we can treat a disease using biotechnology. Today, I want to pick up on how we can diagnose diseases. And the disease that we're going to talk about is Huntington's disease. This disease, it's a neurological disease that causes loss of neurons, typically as people are older, maybe in their 50s, 60s, 70s. And it is caused by a mutation that involves a repetitive area called CAG. Specifically, it's on chromosome 4. And these repeats, CAG, lead to a protein that has glutamine repeated. And when glutamine is repeated enough times, the protein begins to aggregate, and that aggregation interferes with neurons communicating with each other and causes the phenotypic expression or effects of Huntington's disease. What we can do then is try to figure out whether somebody has these CAG repeats. And the key seems to be, if you have 40 of these CAGs in a row, that will lead to Huntington's disease. If you have lower than 40, it's low enough that you won't get Huntington's disease, somewhere about 40 CAG repeats. So in the test, we just need to figure out how many repeats a patient has. Remember, when we discussed mutations and diseases, this is a disease that is called dominant. If you have one copy of a gene, an allele, that has 40 CAG repeats, that is enough. A good copy does not save a bad copy in this case. So let's introduce a technique which is called electrophoresis. Electrophoresis is a process where we can separate out pieces of DNA. We use something called an agarose gel that looks like something like this, and we can see it in here as well. And at the top of the gel, we have little wells, and those little holes allow you to put a sample from a patient in the gel. And you cannot see, but throughout this agarose gel, there are tiny, tiny pores that allow either DNA, RNA, proteins to move through the pores and through the gel. Now, what we do is we take a person's cell, right? And we go inside that cell and we take out the DNA, if that's what we're looking at, which we are in this case. And we chop that DNA up. And when we chop it up, that you get many different sizes of DNA. For simplicity's sake, let's pretend that if I use restriction endonucleases and I chop up DNA, I'm going to get 3 different sizes of DNA. Okay, and so I'm in a test tube. It's in a liquid form. I can't see these, but we're going to add these to the well of the gel. And then we're going to apply a current, and that will pull the negatively charged DNA through the gel. The smaller pieces move through the gel quicker. So if you look at the pieces we'll look at the completed one here, that are further away from where we started, which is up here, they are the smaller pieces. So the shorter ones move faster. They tend to be further from the well. Let's take a look at how we specifically can use this with Huntington's disease. This is a gel, and in this case, we're looking for the CAG repeats and how many of the repeats that you have. So first, what they do is they take the DNA out of the cell, like we said. They're also going to do PCR And PCR is going to allow them to get lots and lots of copies of that particular area on chromosome 4. Now, I want you to focus first on the controls, and these two lanes here, we will load not a patient's DNA, but we will add DNA that we know has 18 CAG repeats. In this lane, this is called a lane, we will add what is known as 48 CAG repeats. This gives us a sense of how big the DNA is going to be of the patient. Now, the other wells, in this case, each of the wells or lanes is loaded with one person's DNA, one patient's DNA. So you can see across here, we have 28 different individuals. And we see a lot of times we'll see two different bands because you remember you have an allele from your mom and an allele from your dad. Now, if we look at patient 1, we see that they have one allele that is about the size of 18 CAGs. And so we call that normal Huntington allele. Remember, at under 40 is what's good. And then this up here, we see one that is approximately 48. You can see they're not all perfectly 48 or 18, but it gives us an idea of what the cutoff, which the cutoff is somewhere around here. So this patient does have the Huntington allele and eventually will develop Huntington's disease. So will this individual, you can see all of these. If we go to individual 5, they have two alleles, both of which are around 18, below the cutoff. So they have two good alleles. They will not develop Huntington's disease. This is how we can use, one way we can use electrophoresis to diagnose a disease. There are many other ways, but I do want to point out that not only is this electrophoresis, but it's also using PCR. PCR was mentioned during our Collaborate session. It is a way to make copies of DNA. So if I just had this one piece of DNA, I would never see it on this gel. No, however you can visualize DNA, one piece would not be enough. And I need multiple. So what I do is I put this piece in a test tube, and in that test tube, I add other reagents, other proteins, and this copy, this one strand will be made into 2, and then that 2 will be made into 4 and 8 and on and on. I will discuss PCR in detail in another lecture. Thank you very much.